Hey everyone, how you all are doing? I hope you all are doing really well as always. So today we're going to talk about types of staining. And we have mainly two types of staining, positive staining and negative staining. We call any type of staining as positive staining when the background remains unstained and here only specimen is stained. It includes simple staining, differential staining which differentiates between cells like we have acid-fast staining, gram staining, and we also have structural staining to stain special structures like flagella, endospore, capsule. And the next we have is negative staining. It is highly specific for a structure in the sense that only some special structure will require negative staining, else positive staining will do. But what exactly happens in negative staining? Here the background is always stained and the specimen might or might not be stained. I want you to look at these two slides. These are the examples of positive staining and this slide is an example of negative staining. Here if you can see, we have stained background and the cells are not stained. But if you look here, here we have unstained background but we have crystal violet stained cells, we have purple cells, we have red cells. These two are examples and one more which is structural staining is an example of positive staining and this only is a negative staining. Now let's look at the simple staining, the first type of staining under positive staining. It is also known as monochrome staining because mono single chrome color. So we're using only one type of stain. Now this type of staining is very basic. Here you look only at if organism is present or not, and if they are present, are they bacteria or are they yeast, and if any one of them, what kind of colony characters are they showing, what shape are they showing. It gives us a very basic, a general idea about organisms present in the sample. The requirements for any simple staining is you need a sample, slides and stain, and that's all you need. I want you to look at the procedure. First of all, of course, you'll prepare a smear, which is air drying, fixing, which we have already discussed in the previous slides and all the previous videos. And then you flood the slide with stain or you add the stain for 45 seconds to 1 minute depending on the type of stain you're using. Then we wash the slide or rinse the slide so that the excess stain gets drained off, and you finally air drying by keeping the slide in the air for some time so that the water and other type of stains get evaporated, and then you finally observe it under the microscope so that you can see the different shapes of organisms. If you look at the resultant slides of simple staining, you will see a white background, right? You are seeing a white background over which there are some stained cells. Now these stained cells are stained on the on the basis of stain that you have used. If you have used crystal violet, these will be violet cells. If you have methylene blue, if you have used methylene blue stain, then you have blue cells. If you have used saffron stain, then you will have red cells. And what is the purpose or advantages of using simple staining? It basically reveals morphology, as we have discussed, the shape, the size of the cells, because you're just looking at the overview of the cells, or also it shows the colony characters, that is how the cells are arranged in the slide, and that is your colony characters. Finally, we have differential staining. Differential staining is a second type of staining under positive staining, and it is differential because it differentiates two bacterial cells or two parts of the cells on the same slide. It differentiates on the basis of different staining reactions, that how a cell or a part of cell is responding to a stain. They are responding differently because of their different molecular composition. I hope that it's clear that it helps us differentiating two bacterial cells or any two parts of the cells on the same slide. Here we are using two stains minimum two stains are used, And the major advantage is to differentiate the bacteria. Like in gram staining, it clearly demarcates two different types of cells, that is gram positive bacteria and gram negative bacteria. Again, the two most common methods are gram staining and acid fast staining, but we have other type of staining under differential staining as well. Now this is the last slide which shows the procedure for differential staining. But I don't want you to look at the procedure, I want you to look at the requirements. Here we have used two different type of stain, if you can see. Here we have crystal violet and here we have used saffron. We have also done decolorizer. We have also done decolorization by using decolorizer. Also we have used mordant. So when you look at the final slide, you will look at two different type of cells stained differently due to two different type of stain used. You have purple cells and you have blue cells. With that, thank you so much guys. I hope you understood everything that we have discussed so far. If you do, make sure that you subscribe to the channel so that you do not miss out on any updates. With that, bye bye, take care.